Alright, hi everyone. Today we're going to cover question 5 and onwards. So I'm not going to read through the whole thing because it's quite a lot to read out loud. But this is question 5. Read the passage below. Use the information in the passage and your own knowledge to answer the questions that follow. Alright, let's start off with part A. Suggest what is meant by the term disaccharide. So di means two. So two of what? Two monosaccharides that are joined together. So you do need to know the three sort of categories of carbohydrates. You have monosaccharides, so that's one unit of sugar. So that's something like glucose. So glucose is a monosaccharide. Disaccharide is when you have two monosaccharides joined together. So that'll be something like lactose. And we can see here, lactose is a disaccharide, which is broken down into glucose and galactose. So glucose is a monosaccharide, galactose is a monosaccharide. So the other third category of carbohydrates that you need to know are polysaccharides. So poly means many. So examples of polysaccharides would be starch or glycogen, for example. All right, so let's write this down. So just what's meant by the term disaccharide. It is a type of sugar where two monosaccharides are joined together. Okay, part B, give a reason why lactose is not absorbed by the epithelium. So let's see what we have here. Oh, it just says lactose is not absorbed. So part of the reason why it's not absorbed is perhaps it could be too large to be absorbed because it needs to get broken down first before it gets absorbed. So let's write that down. Or lactose may be too large. All right. Part C1, explain what is meant by a gene being switched off. So when we say a gene is switched on, then that means that that gene is undergoing transcription and translation. So that just means the DNA will get transcribed to an mRNA, which would then get translated to a protein. So when we say it's switched off, then that means that that gene is not being transcribed and not being translated into a protein. So this means that the gene is not transcribed to form mRNA, and therefore the protein is not produced. Okay, let's move on to part 3. Describe the effects on a person when the gene for producing lactase is switched off. So let's go to this part here. So it says here on the 4th line, so lactase is an enzyme that basically breaks down lactose into glucose and galactose. Right, and then it says here, when the gene is switched off, the person becomes lactose intolerant, which means they cannot digest any of the lactose. So when this gene is switched off, they can't digest the lactose, which means they can't absorb the lactose. So we can write is switched off. Lactose cannot be digested. And therefore cannot be absorbed. Okay, let's move on to part D. Suggest how people in some parts of the world have evolved tolerance because of their diet. So this is a suggest question, which means it's not specifically part of your syllabus, but you do need to use what you have learned and apply that knowledge into this question. So it says how some people in parts of the world have evolved tolerance because of their diet. So it could be because they have or they ingest a lot more dairy or milk into their diet, and the ones that are able to digest it have a better time surviving, right? And that would give them an advantage, and they would pass on these alleles to their offspring. So we can say their diet consisted of a lot of dairy, and individuals that are able to digest lactose better chance surviving and would pass their alleles to their offspring. So I've included some extra things that aren't necessarily part of the marking scheme, but I think it's always safe to just add this into your answer just because it's talking about natural selection. So the 2 points that are in the marking scheme are they would eat a lot more dairy, and then the ones that are able to digest the lactose would give them an advantage. So that would be the 2 marks. Okay, let's move on to part E. Explain how the symptoms of lactose intolerance are caused. So the questions are asking how the symptoms of lactose intolerance are caused. So let's go back up here and let me just highlight this part here where it says at the bottom, symptoms of lactose intolerant include diarrhea, calcium deficiency, and vitamin D deficiency. So we do have to include all 3 of these symptoms. And then in the previous paragraph, so this part here, it says that any lactose that will be taken in the gut will be collected and fermented, forming methane and carbon dioxide and other soluble products. These soluble products lowers the water potential in the gut. So let me include a little diagram for you. So on the left here, we have normal lactose digestion. You can see that the lactose gets broken down into galactose and glucose and then taken up by the small intestine. Whereas on the right, we have lactose intolerance. So what happens is that the bacteria will feed off of the lactose, and that produces things like methane, carbon dioxide, and that lowers the water potential of the gut. So what happens here? You can see that water is going to move from a region of high water potential to a region of low water potential via osmosis. So it'll move from the cells into the small intestine, and that is what causes diarrhea, because diarrhea is just basically watery stools, right? And because you would have these watery stools, or because you have diarrhea, then that would affect the absorption of vitamin D and calcium. So let's write that down. I'm just going to remove this. Okay. So we can say the products of the bacterial fermentation causes the water potential in the gut to decrease. Therefore, is to move via osmosis from the cells into the lumen of the small intestine, or also the gut. So the lumen of the small intestine is the gut in this case. This causes diarrhea, which could lead to poor absorption of calcium ions and vitamin D. Especially because calcium ions are soluble in water. Okay, so in the marking scheme, it does imply that vitamin D is also water soluble, but actually vitamin D is fat soluble. So I'm not really too sure how the marking scheme would fit into this. So let's go through the marks. Water is going to move from the cells, that's one mark, into the lumen of the small intestine, that's the second mark. This causes diarrhea, which leads to poor absorption of calcium ions and vitamin D, that's the third mark, because they are soluble in water, that's the fourth mark. So in the marking scheme, they do imply that vitamin D is water soluble, although vitamin D is actually fat soluble, so I'm not too sure what the marking scheme is about here. All right, let's move on to question 6. Blood will clot when there is damage to the skin, such as a cut. State 2 reasons why this is important for the blood to clot. Okay, so 2 things. You want to prevent things from leaving and you want to prevent things from entering. So in this case, you want to prevent excessive blood loss. And you also want to prevent pathogens or bacteria from coming into the body. So number 1 is to prevent excessive, that's supposed to be a C. Prevent excessive blood loss and to prevent pathogens from entering the body. Okay. Part 2, the diagram shows some of the stages in the process of blood clotting. Component V in the blood. Okay, so you do kind of just need to remember this. So there are several components in the blood. You have your plasma, your red blood cells, your white blood cells. So your red blood cells transport oxygen, your white blood cells help fight against pathogens, and then you have your platelets. So that's component V. So V is the platelets. Okay, let's go on to substance W. So substance W is going to convert this to, so prothrombin to thrombin, and the only thing that can do all of this converting is an enzyme, and here we have this enzyme here. So we can write thrombokinase enzyme. Okay, and then thrombin here converts substance Y into substance Z, and substance Z is the one that forms the clot. The ones that form the clot is referred to as fibrin. So fibrin is insoluble in water, but fibrinogen is soluble. So this would be soluble in the blood. This is insoluble, so that's why it's able to form a blood clot. So this would be fibrinogen. Okay, let's move on to part B. Hemophilia is an inherited condition where the blood of a person does not clot. The diagram shows the family pedigree. Some members of the family have the condition. Deduce the genotypes of family members 1 and 2, showing how you obtained your answer. So for your syllabus, you do need to know 2 examples of sex-linked genetic diseases. One of them is hemophilia, the other one is red-green color blindness. So for sex-linked diseases, they are found on the X chromosome. I'm just going to write this here. And if a person carries, let's say, this allele here, so it's a recessive allele on the X chromosome, then this is the hemophilia allele. So I'm just going to write that down. And then this would be an example of a normal allele. So if we look at family member number 1, so family member number 1 is a hemophiliac male. So because they're male, then that means they are an X, or they carry the XY chromosome. And because they have hemophilia, they would carry the recessive allele on that X chromosome. Now for number 2, family member number 2, we can see that they are a non-hemophiliac female. So if it's female, this person should have 2 X chromosomes. And so if they're non-hemophiliac, there are 2 possibilities here. Either they could be, let me see if I can zoom in a little closer. Either they could be XX and carry the normal allele, or they could carry one of the recessive alleles. So to determine whether or not family member number 2 is homozygous dominant or heterozygous, we can look at the children. So I'll give an example here. So their son has hemophilia. Their son would have an XY chromosome, and because they have hemophilia, they must carry the recessive allele on the X chromosome. I'm just going to do this in a different color so you can see it a bit more clearly. So when it comes to this, right, the Y chromosome is going to be inherited from the father, whereas this X chromosome is going to be inherited from the mother. Then that means that family member number 2 must have the recessive allele. So therefore, let me just write this down properly. Therefore, family member number 2 is a carrier. So she would have both the dominant allele and the recessive allele. So she's heterozygous. Let me just erase all of this. Okay. Okay, so let's scroll down. So we would say the hemophilia allele is located on the X chromosome. Okay. Family member 1. Must be XHY because it's a fail. Family member 2, so the mother, family member 2 must be heterozygous because some of her children allele. Okay, let's go through the marking scheme for this. So located on the X chromosome is one mark. Male must be, or family member 1 must be this, and family member 2 must be XHXH, or heterozygous. The allele is recessive, and it shows up in the next generation. So you mentioned that her children has inherited it. Okay, let's move on to part 2. So male 3 and female 4 have a second child. Deduce the probability that this child will be a boy with haemophilia, showing how you obtained your answer. So let's scroll up for this. So male 3 and female 4. So it's this couple over here. So 3 and 4. So let's zoom in. So let's do this in a different color. I'm going to deduce what the genotype is. Let's start off with the mother. So the mother has 2 X chromosomes, and because they are a hemophiliac, then that means they must carry both of the recessive alleles. Now for the father has an XY, has the XY chromosome, and because they are not a hemophiliac, then that means they must carry the dominant allele on the X chromosome. So you can actually do a little Punnett square with this. So I'm just going to draw it at that question. So I'm just going to draw it in this space here. So male 3 was this, and female 4? Was this. So we can try and draw a little Punnett square to show our working out. You don't have to write this out because they don't specifically ask for this in this question, but sometimes they might ask you to draw it out. So let's just fill this up. So here, these are the daughters and these are the boys, the sons. So they're specifically asking for sons. So there is a 50% chance of getting a son, and then there's a 100% chance that that son would have hemophilia. So 0.5 times 100% chance, that would be 0.5. So there's a 0.5 probability that they will have a boy with hemophilia. So let's write this down here. The female so genotype, right, because they have hemophilia, whereas the male has a genotype the probability of boy is 50%, and the probability of son having hemophilia is 100%. So therefore, the ability with is 0.5 times 1, so 50% times 100%. All right, because they do tell you to show your working out, show how you obtain your answer. All right, let's move on to question 7. The diagram shows a kidney. So which of these is the name of structure M? Structure M is the ureter. Let's do a little bit of recap here. So these two would either be the renal artery or the renal vein. And if you recall, I'm just going to draw like the little kidneys first. So you have these tubes that would then connect to the bladder. Sorry, this is a terrible drawing. And then this would connect to the outside. So this is the bladder here. So these two tubes are referred to as the ureter. And then this, which transports urine from the bladder to the outside world, that would be the urethra. Okay. So part 2, which of these structures are connected to structure M? So I've already drawn that there. That would be the bladder. So the bladder is responsible for storing urine. Part B, state 3 differences between the blood entering the kidney and the blood leaving the kidney. So because the organ consists of cells and cells undergo respiration, that means that the blood coming into the kidney would have more oxygen, and the blood leaving the kidney would have more carbon dioxide. So we can write that down. So entering kidney would have more oxygen. The blood leaving the kidney have more carbon dioxide. Another thing as well is that your kidney is responsible for filtering out urea from your blood. So the blood leaving the kidney should have less urea. So less urea. Okay, part C, the table compares the nitrogen content of urine during 24 hours for a person on high protein diet with a person on low protein diet. So for this, we want to find out the difference between these two and then divide it by this value here because it says compared with the low protein diet. So we would do 16.8 minus 3.6 and then divide it by 3.6, and because it's a percentage difference, we would times that by 100. So let's calculate that. So 16.8 minus 3.6, divide that by 3.6 times 100. That should give us 366.67%. So 366.67%. So it is more than 100%, and we can see here that this number is much smaller than this number over here by almost 4-folds. Okay, so let's move on to part 2. Which is the main nitrogen-containing excretory product produced from the breakdown of protein in the diet. Use the information in the table to explain your answer. So let's go back up to the table. So when a person eats a lot of protein, and we can see here it goes from 3.6 grams to 16.8 grams, a large percentage of that actually gets converted into urea. So out of the 16.8 grams, 14.7 grams of it would be urea, and that's a large increase from 2.2. So let's write that down. So urea is the main product as a large increase of urea seen comparing the low diet to high protein diet. All right, and this should be the last question. So part 3, the breakdown of muscle tissue continues at a constant rate and is not affected by diet. Discuss whether the information in the table supports this statement. So let's scroll up to the table here. So if we compare the high protein diet versus a low protein diet, we can see an increase for urea, an increase for uric acid, an increase for ammonia. But if we look at creatinine, it's more or less the same. It's 0.58 versus 0.6. So that means that this table does support the statement. So we can say, when comparing the low protein diet with the high protein diet, the it's mass, right? It's in grams, yep. Mass of urea, uric acid, and ammonia increase. However, with creatinine, the masses in both diets are quite similar. The reduction of creatinine could be due to the breakdown of muscle tissue. All right, this is the end of paper 2R. Let's go over the marks for this. So 1 mark for saying that there is a difference in mass of urea, uric acid, and ammonia when comparing the two diets. That's 1 mark. Another mark for reference to creatinine and how they are quite similar. So that's actually 3 marks right now. In both diets, that's actually 4 marks now. And so this could be due to the breakdown of muscle tissue. That's 5 marks. All right, that's the end of paper 2R.